This is one of several videos that I've made about my research on the biology of the Florida harvester ant. The Florida harvester ant, Bergonomyrmex badius, is a large ant of the sandy coastal plains of Florida. It gets its name from the fact that it collects large numbers of seeds and stores them in the nest and chambers for later consumption. This video is about some of the more surprising and interesting features. Hunting and gathering has been part of the human food strategy since early in human evolution. The same food strategy has evolved in a number of ant species, but it did so tens of millions of years before it appeared among humans. In many ant species, including this black harvester ant from the California desert, this means a combination of predation, scavenging, and collecting seeds. The Florida harvester ant, Bergonomyrmex badius, is a charismatic ant of the sandy coastal plains of Florida, Georgia, and South Carolina. Its nests are made conspicuous by the fact that the ants collect bits of charcoal to decorate their nest disc, creating a black disc that contrasts with the white sand. Foragers roam the territory to hunt insect prey and to collect seeds for consumption or storage in underground chambers. The ants use the seeds by chewing them into ant bread, which they feed the larvae. Chewing begins the malting process and aids the digestion of the seeds. The seed stores can be readily exposed by excavating a nest, chamber by chamber. Here I'm using a shop vacuum to expose an underground seed chamber.
So we've exposed the seed chamber here. There's a few seeds in this one side. When the ants have really, when they store a lot of seeds in the chambers, then the chamber can be completely full of seeds, so that there's only just space for an ant to crawl between the top of the seeds and the ceiling.
This chamber is almost filled with seeds. The exit entrance to the shaft is at the top, and the workers are circled in red. A nest may contain 1 to a dozen of these seed chambers. Colonies store huge numbers of seeds. For example, this colony of 7,500 workers had stored almost 100,000 seeds weighing 300 grams. These are relatively large seeds. This small colony of only 900 workers had 11,000 seeds weighing 11 grams. Here, 3,600 workers collected 250,000 seeds weighing 205 grams. These are relatively small seeds. And here, only 900 workers stored 38,000 seeds weighing 45 grams. Every seed represents a minimum of 1 trip by a forager. Colonies store between 15 and 35 seeds per worker, amounting to 20 to 50 milligrams. This ratio is the same for all colony sizes. At Ant Heaven, the ants collected the seeds of over 50 species of plants, with fewer than a dozen making up most of their numbers and weight. The seeds were identified and photographed by my assistant, Julio Dominguez. And here are some of the prettier ones. Not surprisingly, the seeds vary enormously in size, with the largest weighing almost 90 times as much as the smallest. Here the species are ranked by size. A look at the relative sizes of the ants and the seeds they collect makes one question whether the ants can actually open the larger seeds. To test whether the ants could crack open and eat larger seeds, we offered lab colonies 4 sizes of seeds, each marked with a different color of fluorescent ink, shown under ultraviolet light here. Lab colonies brought all sizes of seeds into their nests. But they were able to open and eat only the smallest seeds. Major workers are considered to be seed millers, but although colonies with major workers husked more seeds, they were no more successful in opening the larger seeds. Were field colonies also unable to open large seeds? We offered field colonies the same 4 sizes. Over a couple of weeks, only the husks of the smallest seeds appeared in the trash heap. Analysis of the colony middens showed that most of the seed husks were of much smaller seeds, primarily grasses, than brought in by the foragers, which is the blue line. Analysis of the seeds stored by over 30 colonies showed that the majority of the seeds were actually too large for the ants to open, are shown in pink. This amounted to about 70% of the seeds in storage. The axes are reversed here, by the way, the size decreasing from left to right. Interestingly, when the ants relocate their nests, they move all of the seeds, including those too large to open, into the new nest. My assistant Julio Dominguez took these seeds from workers, transporting them from the old nest to the new. It seems counterintuitive that ants expend so much time collecting and maintaining a resource they apparently cannot use. Add to this that the collection of this resource risked the lives of thousands of foragers whose life expectancy was therefore only about 3 weeks. How could such an apparently unrewarded behavior evolve? In trying to find an answer, I initially tested what later turned out to be the wrong hypothesis, namely, what kept the seeds from sprouting. Christina Kwapich and I buried seeds in plaster-bottom petri dishes and found that in the absence of ants, some seeds germinated. Did this confirm that ants prevent the seeds from germinating? But what if this thinking was backwards? What if the ants actually wait for the seeds to germinate and then eat the vulnerable exposed contents? We tested this idea both in the lab and in the field by staining germinating seeds with fluorescent dye rhodamine B. But as a control, it was also necessary to dye the insides of non-germinating seeds a different color. To do this, I drilled tiny holes in the seed coats and soaked the seeds in methylene blue, a vital dye. The holes were then sealed. The seed flesh was stained a dark blue. When we fed both such dyed seeds to lab colonies, the larvae quickly became pink and fluoresced brightly under UV light. The proportion of larvae that had eaten pink seeds peaked in 2 days. Essentially, no non-germinating seeds were eaten, the green line. Feeding these dyed seeds to field colonies gave the same results, with only germinating seeds being eaten. But do the seeds actually germinate in the chambers, and do the ants then eat them? To test this, Julio Dominguez and I dug pits next to the nests, cut into the seed chambers from the side, and cut the chamber in half with a metal barrier. This cut off the ants' access to part of the seed chamber, so they could not remove germinating seeds. Excavating such chambers after a week or two showed 16 times as many germinating seeds on the access denied side of the barrier. The ants had removed and presumably eaten the germinating seeds they had access to. This is strong evidence that the ant strategy is to store seeds too large to open, wait for them to germinate, and then eat them, a sort of wild farming. The seeds of wild plants do not typically all germinate at once, but form seed banks in the soil of which only a fraction germinate each year, and possibly only under certain circumstances. To estimate the payoff of this strategy, we needed to know how the various seed species respond over time and with seasons. As a proxy for species, we sorted seeds into 4 size classes. The pink ones are those the ants cannot open. We placed them into plaster-bottom petri dishes and buried a set of 4 at 4 depths every 2 months for 1 month. We also kept a duplicate set of seeds simultaneously in the lab at 4 different temperatures. This complex experiment, with all its attention to detail and schedule, was carried out by my assistant Nicholas Handley. The germination rate is shown on the y-axis and the burial date on the x-axis. The blue band is the soil temperature. Though the germination of the largest seeds, blue line, and the smallest, the pink line, gradually increased over the year, more strongly so for the smallest seeds. In contrast, the middle 2 sizes showed strong peak of germination in the spring and fall and very low germination in midsummer. This suggested that both temperature and time affect seed germination, with high summer soil temperatures inhibiting the germination of the middle 2 sizes. At the same time that we buried the seeds in the field, we also tested the same 4 sizes at 10, 14, 24, and 32 degrees in the lab. Germination differed by seed size and temperature. The lab patterns help explain the field observations as follows. The very large seeds, shown in blue, germinated more at higher temperatures and almost not at all at 10 degrees, but this improved later in the year. This corresponds to their gradual increase in germination in buried seeds over the year. The large seeds, red, initially germinated best at 10 and 15 degrees, but as the year went on, they germinated only at 15 degrees. As a result, their germination was greatly reduced in the summer when soils were warm. The medium seeds, green, were less responsive to temperature. Initially, they germinated more at lower temperatures, but after August, they did better at the higher temperatures. As a result, their germination was greatly reduced in the summer. The small seeds, in pink, changed from initially germinating less as temperature rose to germinating similarly at all temperatures by October. This corresponds to their gradual increase in germination in the buried seeds over the year. Seed size is not totally congruent with species. However, the results for seed size were greatly dominated by seeds of a few species, as shown here. Thus, results greatly overlap with the species. How can we understand these diverse findings? Harvester ants are gambling on the future possibility of being able to eat their stored seeds when the seeds, germinate. The payoff for this gamble depends on the seed species, the season, and the elapsed time. Here's a way to understand the gamble. If the ants consume, say, 30 small seeds in the time it takes a large seed to germinate, then the gamble pays off if the large seed weighs more than 30 small seeds. In this way, the ants have increased their resource base many-fold over only depending on seeds they can open. But the payoff of this gamble is not assured. Each panel here is the seed size distribution within a colony. The great differences in the distributions means that the payoff for each colony will be quite different. The differences probably result from differences in the patch of habitat the ants occupy, as well as differences in worker preferences for seeds. The lives of harvester ants are a reminder of how much all lives, including ours, are dependent on chance. All creatures make decisions that are gambles because the future consequences of these decisions are unknown and unknowable. This capriciousness binds all living things. It's a fundamental truth of life for us as well as harvester ants.